Gene cloning in eukaryotes. Gene cloning have been carried out mostly in bacteria. But many difficulties are associated with them, for example, 1. Correction of introns of eukaryotic mRNAs, 2. Failure of transfer of equal number of plasmids into daughter cells during cell division and yield of two types of cells, one with plasmid and the second without plasmid. Post-translational modification, e.g. inhibition of proteolysis, addition of oligosaccharides to specific sites on the polypeptide chain, the glycosylation 3. Threat for hazardous effects and 4. Expression to ensure for the presence of a stable plasmid in the bacterium when applied on large scale for commercial applications. With these prospects, the attraction of the use of eukaryotic cells is obvious. In recent years, considerable efforts have been made for the improvement of crop plants through genetic engineering. Many works on various aspects of building up of vectors and expression of insert genes in eukaryotic cells are in progress. Some of them are briefly described as below. 1. Fungal cells. There is a large number of plants ranging from unicellular to multicellular forms. Success has been achieved in some plants. A. Yeasts. Yeast is an unicellular eukaryotic fungus containing a small, well-characterized genome. Unlike plant or animal cells, it has rather fast growth rate and itself is a non-pathogenic fungus. Most of its genes contain introns which are spliced during purification of mature mRNA. It appears that introns found in yeast contain sequences for correct splicing as they are totally absent in higher eukaryotes. Moreover, yeasts can carry out post-translational modifications such as removal of a signal sequence from a precursor polypeptide after the secretion of cells. This reveals a major advantage of yeasts over the bacteria. Success in DNA cloning in yeast depends on uptake of foreign DNA by its spheroplast. Naked protoplast. One cell wall is enzymatically removed in the presence of Ca++ and polyethylene glycol PEG. The spheroplasts develop cell wall after the incorporation of DNA. B. Transformation in filamentous fungi. Transformation through plasmid of E. coli was first described in protoplasts of S. cerevisiae and then in N. crassa. However, the situation in filamentous fungi is different from the others as there is no convincing evidence for autonomously replicating plasmids. But sequence having functions similar to ours have been isolated. Method of transformation is similar as for plant cells. Following are the examples where transformations in protoplasts have been carried out: anidulins, A. niger, or otizi, Cephalosporium acrimonium, Coprinus cinereus, Glomerula cingulata, Guiamanomyces graminis, Mucar sp, Penicillium chrysogenum, Septorium nodorum, etc. 1. Application of transformation of fungal protoplasts. The transformation approaches are used, 1. For the analysis of promoters and construction of expression vectors, and 2. Biotechnological application in the expression of foreign genes in fungi. Analysis of promoter involves the construction of vector which have different pieces of promoters. These can be cut by restriction enzymes and joined with DNA ligase. By this method, changes in promoter region are also possible, and this can be achieved through mutagenesis. Thus, a strain can be improved resulting in overproduction of the products. Adopting these approaches, many mammalian genes have now been expressed in S. cerevisiae and aspergilli through expression vector, i.e. vector which include sequences promoting high level of transcription of the gene and secretion of the gene products. In the USA and Europe, several companies have demonstrated the expression of foreign DNA introduced in Aspergillus sp. through transformation. Upshall et al. 1987 have constructed a plasmid PM159 for the secretion of human tissue plasminogen activator by anidulins. Yield of this protein was about 100 mg liter culture filtrate. Similarly, D. M. Gwynn and collaborators in 1987 found the secretion of human interferon by transformed anidulins mycelia to about Img liter. 2. Plant cells. There are large number of plants which have been selected for transformation of cells by using different approaches. A. Gene transfer in dicots by using Agrobacterium Ti DNA is vector. Transfer of T plasmid into plant cell is not possible because of its large size, most probably up to 235 kilobits. Therefore, is excised and used as foreign DNA for expression in plant cells. From bacteriological point of view, apine production and development of gall are not required for the successful integration and expression of DNA. Hence, replacement of nose and ops genes with a foreign DNA fragment retaining the apine synthase promoter for expression of inserted DNA fragment has no harm. In 1983, J. Skell and M. Van Montague undertook some experiments where genes for resistance to kanamycin and methotrexate expressed in cultured callus cells. Generally, it is easy to use apine promoters for the expression of foreign DNA. It has become possible that the inserted genes, like other plant genes, will express only at certain stages or specific tissues of plants when they are with specific regulated promoters. There are reports of replacement of apine synthase promoters by regulatory promoters obtained from plants. N. Murray and co-workers in 1983 have introduced gene for bean plant storage protein, the phaseolin, which is supplemented with methionine codon into the cells of sunflower plant after transformation with T plasmid, where phaseolin gene was under the control of its own promoters, Thi octopene synthase. For details see chapter 13. B. Gene transfer in monocots. Thi, T plasmid is specific only to the dicot plants. Autumnfacines does not induce tumor in monocot plants, however, most of the major crops including cereals are monocots. G.M.S. Hoykas Van and co-workers in 1984 discovered that autumnfacines could transfer into certain monocots, resulting in expression of apine gene within the plant cell without inducing the tumor. This discovery made it possible that can be expressed into cells of monocots. C. Plant cell transformation. Plant cell suspension culture coupled with DNA transfer techniques has become a new field of gene manipulation in plants. Cell suspension cultures have been the traditional source of plant material for biochemical selection of plant cell mutants, such as numerous cell lines resistant to various amino analogues. Suspension of protoplasts constitutes an identical plant material from one laboratory to other. The approaches made for plant protoplast transformation are: 1. Introduction of DNA sequence via integration into T plasmid of autumnfacines. 2. Transfer of naked DNA into protoplast including bacterial spheroplasts and microinjection. And 3. Transformation with plant viruses carrying inserts, e.g. Gemini and colimaviruses. The plant protoplast treated with PEG, used for protoplast fusion, resulted in efficient delivery of DNA to protoplasts. The PEG method has been employed for the successful transformation of protoplasts with T plasmid. Once DNA fragment is introduced into plant cells, it integrates with plant chromosomes and expresses from its signal. Transformation of plant cells with a gene for drug and antibiotic resistance linked to plant promoter has been possible. The gene could show expression in plant cell. Therefore, the transformed cells could be selected from the culture medium amended with drugs and antibiotics. The plants regenerated from cells show drug antibiotics resistance. 1. Plant cell transformation by ultrasonication. Ultrasonication has been done for successful gene delivery. The Biotechnology Research Center, Beijing, China, has used this technique for gene transfer in plants like wheat, tobacco, and sugar beet. When the cultured explants were sonicated with plasmid DNA carrying marker genes such as CAT, NPTLL, and GUS and sonicated Cali transferred to selective medium, shoots were grown successfully. The Cali in control set of experiment which were not sonicated with plasmid did not grow on selective medium but they died. Transgenic tobacco plants were obtained at 22% frequency. 2. Liposome-mediated gene transfer. Liposomes are small-sized particles containing a phospholipid bilayers which are concentric in nature. Liposomes enclose a aqueous chamber and can entrap water-soluble molecules. Therefore, they are called lipid bags. Many plasmids are enclosed in them. By using polyethylene glycol PEG, they may be stimulated to fuse with protoplast. In several plants like carrot, tobacco, petunia, etc. This technique has been used for transfer of genes. Due to endocyclosis of liposomes, DNA enters the protoplast. Success: it gets adhered first to protoplast surface and get fused with protoplast at the site of surface. Consequently, plasmids are released inside the cell. There are several advantages in using this technique such as 1. Low toxicity, 2. Protection of DNA RNA from nucleases that lies them, 3. Long stable storage of DNA fragments in liposome, 4. Applicability in various cell types, and 5. High level of reproductivity. Note: Sunbeet plant. Scientists at Agrogenetics Corporation, USA, are concentrating on phaseolin gene, which is a major protein of French bean. It has low methionine content owing to which food value of the bean is reduced. Phaseolin gene was successfully transferred to cultured cells of sunflower. The callus-producing bean protein is called as sunbean. The phaseolin gene can be put back into bean plant. By doing so, the nutritional value of the crop can be enhanced. 3. Electroporation. Electroporation, using electric field, is used to transfer the foreign DNA via the fragile cells. Brief pulses of high voltage electricity, about 350 volts, is applied to protoplast suspension containing naked or recombinant plasmids. The electric pulses induce the formation of large pores in the cell membrane. These pores give a passage through which the foreign DNA can enter into the protoplasts and thus increase the transformation frequency. The transformed protoplasts are cultured for about 1 month. These develop microcalli, which are solid medium containing selective marker, e.g. kanamycin. After 37 to 45 days, the calli plan are analyzed for the presence of differences in transformed cells. By using electroporation method, genes of choice have been successfully transferred in protoplasts of wheat, rice, petunia, sorghum, maize and tobacco. Moreover, transformation frequency can be improved by using linear DNA instead of circular one, by giving heat shock to protoplast at 45 C for 5 minutes, by adding PEG and using 1.25 kV per centimeter voltage. 4. Particle bombardment gun. It was developed by Stanford and co-workers of Cornell University, USA, in 1987. As the term denotes, it shoots foreign DNA into plant cells or tissue at a very high speed. This technique is also known as particle bombardment, particle gun method, bioelastic process, microprojectile bombardment, or particle acceleration. This technique is most suitable for those plants which hardly regenerate and do not show sufficient response to gene trans through agrobacterium, for example, rice, wheat, corn, sorghum, chickpea and pigeon pea etc. The apparatus consists of a chamber connected to an outlet to create vacuum, figure 6.16. At the top, a cylinder is temporarily sealed off from the rest of chamber with a plastic rupture disc. Gas flows into the cylinder. A plastic microcarrier is placed close to rupture disc. It contains DNA-coated tungsten particle, the microscopic pellets, i.e. coated microprojectiles. When to work, the apparatus is placed in laminar flow just to maintain sterile conditions. The target cell's tissue are placed in the apparatus. A stopping screen is put between the target cells and microcarrier assembly. Helium gas is flown in the cylinder at high velocity. When pressure of cylinder exceeds the bursting point of plastic disc, it gets ruptured. Helium shock waves propel the plastic microcarrier containing DNA-coated micropellets. The stopping screen allows the micropellets to pass through and deliver DNA into target cells. The transformed cells are regenerated onto nutrient medium. The regenerated plant tissues are selected over culture media containing either antibiotics or herbicide. The selected plants are then analyzed for expression of foreign DNA that has been inserted into it. Scientists have got success in delivering foreign DNA into epidermal tissues of allium sapa, scutellar tissues of maize and leaf and cell culture of many crops. Peters, 1993. In addition to bacterial cells, algae, fungi, plant organelles, e.g. chloroplast and mitochondria, and animal and human cells and fruit fly bryas have been successfully transformed. Conditions for bombardment. It requires several conditions required for introduction of foreign gene into the desired cell tissue, pollen, etc. The CP conditions required for introduction of Sus glucuronidis gus reporter gene. For details see Chapter 13, section Transgenic plants for transformation of basmati indica rice have been given in Table 6.1. D. Manhas et al. 1996 has reported for the first time the transient expression of Gus reporter gene through bioelastic delivery in macho embryo-derived embryonic callus cultures of Oryza sativ CV basmati 370. In 1998, scientists of Plant Transformation Group at ICGEB, New Delhi, have got success in transferring human interferon gamma gene into chloroplasts of tobacco, maize, etc. using a particle gum-mediated gene transformation. See the Table 6.1. Bombardment conditions employed on basmati indica rice callus. 2. Pollen transformation through particle bombardment. This method is time-consuming, requires special techniques, and efficiency of getting stable regenerants is low. S. M. Ramaya and D. Z. Skinner in 1997 produced transgenic alfalfa through direct delivery of DNA into pollen grains by particle bombardment method. Plasmid PB1121 bearing Gus reporter gene was introduced into pollen grains. The microprojectile bombarded liquid pollen suspension was allowed to fertilize the tagged and tripped recipient female flower. Pollinated flowers set seeds in about a month. 30% of plants derived from fertile seeds showed integration of Gus plasmid. It was confirmed by southern analysis. After 10 vegetative generations, some transgenic plants lost the integrated Gus plasmid, whereas in few others copies of Gus gene decreased due to unknown reasons. An outline of production of transgenic alfalfa plant through pollen transformation is given in Figure 6.17. See the Figure 6.17. 3. Animal cells. Animal cell culture on a large scale is more difficult and expensive than plant cells, the bacteria, yeast, etc. This is why the commercialization of natural products of animal origin is limited. However, most of the animal products have been produced by using bacteria as biological tool. In India, K. Chandrasekharan, Center for Cellular and Molecular Biology, CCMB, Hyderabad emphasized the use of recombinant DNA technology. Introduction of foreign DNA into mammalian cells. This will help in curing certain genetic defects. Some of the examples of gene cloning in animals are described in Chapter 7. A. Animal viruses. Several animal viruses are known, for example, simian virus 40, SV40, adenovirus, retrovirus, ssRNA, vaccinia virus, etc. They can increase the efficiency of animal cell transformation if used as vector. A special feature of viruses is that they contain the strong promoters which can possibly bring about expression of inserted DNA fragment of the viruses. The most commonly used virus is SV40, which contains a circular DNA of about 5.2 kilobits. In addition to ORI region, the DNA contains early genes and late genes. Early genes are required for DNA replication, whereas late genes encode protein. The amount of DNA to packed into virus capsid is limited. The genome has 3 identified non-essential regions, hence the use of non-defective viral vectors is limited. The defective virus vectors have missing genes whose infection process has to help by viral DNA constituting functional copies of missing genes. If the COS cells, the host monkey cells, are used as host, they can be transformed by a plasmid carrying the early region of SV40 with a defective replication origin. COS cells express wild-type large T antigen and can the growth of SV40. Recombinants containing foreign DNA inserted into early region can be propagated without the need of helper virus. Thus the recombinant and helper can separately recovered from the host. B. Microinjection. Foreign DNA can delivered into a living cell, egg embryos of animals through a glass micropipette. This technique is called microinjection. One end of a glass micropipette is heated until the glass becomes somewhat liquefied. It is quickly stretched, which forms a very fine tip at the heated end. The tip of the pipette attains to about 0.5 mm diameter, which resembles an injection needle. The process of delivering foreign DNA is done under a powerful microscope. Cells to be microinjected are placed in a container, Figure 6.18. A holding pipette is placed in the field of view of the microscope. The holding pipette holds a target cell at the tip when gently sucked. The tip of the micropipette is injected through the membrane of the cell and contents of the needle are delivered into the cytoplasm. For the study of transcription by microinjection, Xenopus oocytes have been widely used because oocytes contain between 6,000 and or more RNA polymerase molecules than somatic cells. Microinjection is technically easy because of large size of oocytes. Some of the endogenous pattern of gene regulation during development has been characterized. The injected DNA integrates randomly with nuclear DNA, and its expression could be possible only when the foreign DNA is attached to a suitable promoter sequence. There are many examples where different types of animal cells have been microinjected successfully. G. Rubin and Spradeling in 1982 for the first time introduced Drosophila gene for xanthine dehydrogenase into a P element parental element, which was microinjected with an intact helper P element into embryo deficient for this gene. Such embryos later on developed flies with rosy-colored eyes but not mosaic eyes as in the first parental generation. Production of transgenic animals. In 1982, R. D. Parmeter of Washington University and R. L. Brinter of Pennsylvania University isolated the rabbit growth hormone sus globin gene, human growth hormone sus globin gene, as well as thymidine kinase gene, and linked separately to the promoter region of mouse associated with the metallothionein I gene, a gene which encodes a metal-binding protein. This was joined to PBR322 plasmid to produce the recombinant plasmids. Macho eggs from adult mouse were recovered surgically and fertilized with sperms in vitro. Immediately the fertilized eggs were microinjected with recombinant plasmids before the sperm and egg nuclei have fused to form a diploid zygote. The plasmids generally combine homologously with each other within the AG, forming a long repeated chincatema which then integrates randomly to give repeated genes at a single chromosomal site. The engineered embryos were then implanted into the uterus of a host mouse mother for further development. The resulting mice are called transgenic mice, since part of genome comes from another genetically unrelated organism. Due to introduction of foreign gene before nuclear fusion, chromosomal integration takes place early and progeny contains new genes. Size and body weight of progenies were extremely larger than the normal ones. Figure 6.19. See the figure. Figure 6.19. Production of transgenic mice, MT, methionine. Further palmator-injected mouse embryos with a DNA fragment containing the rat hormone gene fused to the promoter region of the mouse metallothionein J gene. In this experiment, linear DNA fragments were used rather than plasmids because these integrate more efficiently into the mouse chromosomes. Consequently, 21 mice were produced, among them 7 contained the fusion gene. 6 were twofold larger in body weight than the others. The level of growth hormones increased many times, between 200 to 800 times more than the control. In addition, the level of growth hormone mRNA was also increased in liver cells. A direct transformation, the mammalian cells, can be transformed by the foreign DNA fragments. Therefore, it is necessary to precipitate the DNA with calcium phosphate and mix the cells to be transformed. DNA molecule passes through cell membrane and integrates randomly with mammalian chromosome. Using this technique, selective marker can be linked with DNA fragment to be cloned and can be introduced into mammalian cells. The transformed cells are, thereafter, separated from cell line after plating them on selective medium. MAN techniques are used to recover the selectable marker gene from the biochemically transformed cell line. Following these biochemical processes, M. Perucho et al. 1980 successfully isolated chicken thymidine gene TK gene by ligating with PBr322 and transforming the TK deficient of thymidine gene mouse. LDNA gene sequencing.